Good morning, everyone. Welcome to Pediatric Grand Round. First, I'm back in person in 15 months, I believe. And here to lead us off is Dr. Konark Mukherjee, Assistant Professor at FBRI at the Center for Neurobiology Research and Assistant Professor, Department of Psychiatry and Behavioral Medicine. Dr. Mukherjee received his medical training in India and went on to receive a PhD in immunology at National Institute of Immunology. He completed multiple postdoc fellowships in neuroscience at the University of Texas, medicine at Stanford, biology at Brandeis, and neurobiology at the Max Planck Institute. His areas of interest are focuses on the role of the Mukherjee protein in neurodevelopment. He is the PI in multiple grants, including those investigating optic nerve hypoplasia and Cas mutation, and currently epileptic encephalopathy. Please join me in welcoming Dr. Konark Mukherjee.
Thank you. So, as we all can relate, I'm really excited to be here, and it's great to have at least personal interaction while giving a talk. For the last year, our life has been Zoomified. And so what I decided, because I can now use my hands and facial expressions to explain things, that instead of launching into facts and trivia about some disorder or something like that, I will go in a format of telling you stories. Now, these are stories, but they are not fictions. They are real stories. And there will be two anti-parallel stories, and then we'll give some data from what we have and tell you what we think of these disorders now. So the topic is essentially how some of this X-linked neurodevelopmental disorder, what we think of them now, could actually be neurodegenerative in their pathology, but we are just missing it and how that can happen. So to begin with, how do I change the slide? Should I turn this off?
Oh, I can just click this one.
Okay.
There.
Okay, great. So, so our first story here starts with this person here, Andreas RETT, a physician in Vienna, in Austria. And in early 1960s and mid-1960s, he made a remarkable observation. Some of these girls would come to his clinic. They have a normal infancy, and the developmental milestones are acquired at the right time. And then they suddenly showed neuroregression. And he started collecting evidence what was happening and published eventually a small paper in this German magazine. Of course, this one is a translation of his original title, on a remarkable syndrome of cerebral atrophy associated with hyperammonemia in childhood. And look at the word, the cerebral atrophy clearly is, in his pathology, is looking at some condition which is degenerative, where basically the cerebrum forms and then degenerates back. And this was published in 1966. But it didn't get really much traction, and there's a reason for that. First of all, post-World War, English was established as the language of science. And this article was written in German, so not many people read it. The second one is that he made a critical mistake. So the lab work were wrong, and there was really no hyperammonemia. So these two problems led to the fact that nobody really read his work back then. But then a few years later, Dr. Hagberg, a Swiss physician again, went through 35 cases, and he wrote this paper, A Progressive Syndrome of Autism, Dementia, Ataxia, Loss of Purposeful Hand Use in Girls, called it RETT syndrome. This is really his academic integrity that he gave Andreas RETT his due acknowledgement, even after so many years, and made sure that it's called RETT syndrome. Soon after this paper was published, physicians world over realized they all see these girls who come to them and apparently have a normal developmental milestones and normal child infancy, and then at some point of time in their life, early childhood, they have this neuroregression. And a parent, Kathy Hunter, a mother here in US, whose daughter had similar condition, what she did with help of other parents, established this International Rett Syndrome Association in 1984 in the United States. And with the shift of funding, the research in this Rett syndrome condition moved from Europe to the United States. Soon thereafter, in 1985, Hugo Moser at Johns Hopkins University actually, for the first time, held a Rett syndrome symposium in the United States. This symposium was attended by likes of Fralin Percy, Huda Zoghbi, and all the stars that we know today of this Rett syndrome condition. And soon thereafter, Percy, Zoghbi, and Riccardi published their paper from US, Rett syndrome, initial experience with an emerging clinical entity. So this was always there. Rett had actually seen this very, very early on, but it took several years to be caught up. So this disorder I'm talking of started by actual discovery of the disease just from clinical and pathological impression. And this group published several very powerful papers, and pathologist Dr. Armstrong helped in their work. And if you look at this early work from their group, this is 1988, that is 3 years after the original paper. If you look at their findings, you'll see this diffuse cortical atrophy with microcephaly and mild diffuse cortical atrophy with increased amount of neuron lipofuscin. Essentially, what this is telling you that their initial findings remarkably parallels whatever Dr. Andreas had found, that it is cerebral atrophy, you have neuronal lipofuscin, mild gliosis, so all the telltale sign of degeneration present here. But then something remarkable happened in 1999. Huda Zoghbi made this famous discovery linking one of the first and the most prominent genes called MECP2, which is an X-linked gene with a condition called Rett syndrome. So her discovery is this watershed moment in terms of biology of the disorder, where we had this first gene that she found, and soon the narrative sort of changed, kind of switched over. And 4 years later, she wrote this review article. She now calls it postnatal neurodevelopmental disorders, and the emphasis is on synapses. Something happened at the synapse, and that's why it's a postnatal neurodevelopmental disorder. And then it got firmly established as one of the most prototypical neurodevelopmental disorder of childhood, because this was written by Huda Zoghbi and Neil. Rett syndrome, a prototypical neurodevelopmental disorder. So you see how it started off as a cerebral atrophy and then came down to the time, and now it's a neurodevelopmental disorder. Now, what happened? What made this switch? Now, once this gene was identified, naturally, biologists, in this case, Dr. Bird, made a mouse which did not have MECP2 knockout mouse, and started studying it. The unfortunate part is mice are very good tools. They're really great models, but mouse are not small humans. Their biology is significantly different. So MECP2 was not as important in mice as they are in humans. So imagine if there's a boy who doesn't have MECP2 because boys have only one X chromosome. It's nearly always lethal in early infancy and late childhood. But for mice, the knockout mice lived up to 6 weeks of age. Now, 6 weeks of age in mice is sexual maturity. And the females who are supposed to have the atrophic microencephaly condition did not develop almost any pathology, and only after 6 weeks showed some behavioral symptoms. So it's fairly mild condition. Now, what would you do with this mice where you don't have a pathology but have some behavioral symptoms? All that you could do is do physiology. So people started doing synaptic physiology, synaptic defects were found, and that's how the focus shifted to synapses. And this concept that is a neurodevelopmental, it's a developmental disorder, started. Now, completely parallel to this story is the story of microcephaly with pontocerebellar hypoplasia. That also happens because mutation in a gene called CASC that's also present in X chromosome. Cask was identified as a LIN2 gene in a worm in a really a strong scientific investigation. In mid-70s, there was this huge drive for us to understand how genes are involved in development of different tissue and cells. And people started doing a lot of forward genetics using invertebrate models like worms and flies and trying to figure out how these genes, if they are lacking, would give rise to a developmental condition. And Dr. Horowitz and Sulston, who ended up getting Nobel Prize for their great work on worms, set up a screen where they would do a forward genetic screen and see how developmental was affected and pick up genes where they felt that the lineage of the cells were altered. They did that by looking at the verbal development, and the genes that they got the nomenclature depending on how they cloned it as LIN1, LIN2, LIN3, LIN4, and LIN5, so on and so forth. They published their first paper on this, identifying the molecule LIN2, which is the Cask ortholog, as early as 1980. So this was not identified as a disorder. This gene was identified in worm as a possible gene involved in development. So this is just opposite of the RETT syndrome. Nothing happened again from 80 to 1996, 16 years, when Hoskins et al. Published this paper, where they realized this LIN2 gene encodes a membrane-associated guanylate kinase protein, megaprotein, with a weird calmodulin kinase domain at the N-terminus. That's what they found. This have bulbar induction deficit. Surprisingly, they did not find any neuronal deficit in this work. There was nothing else apart from the bulbar defect that they could find in this work. In the same year, in Drosophila, this strange calmodulin kinase was discovered, but the story here was also somewhat unfortunate. They also were studying Cask. They just did not know that they're studying Cask. The reason they did not know that they were studying Cask because when they cloned in Cask, this molecule they called Kaki back then, there was a frame-shift mutation in their cloning, and they only got the first domain, which is the kinase domain. So they ended up calling it Kaki, but this is a discovery of Cask in Drosophila. And finally, in the same year, in Tom's lab, they were trying to find what are the scaffolding molecules within the synapse using biochemical assays, using a synaptic adhesion molecule as a base. And they found this molecule, which is a Maguk protein with a strange calmodulin-dependent protein kinase at the N-terminus, and Tom ended up calling it Cask. Now, by the time I came to Tom's lab, Tom had forgotten why he called it Cask. In his entire paper, first paper, he doesn't mention why he's calling it Cask. And somebody in the scientific community took upon themselves to call it calcium calmodulin-dependent serine protein kinase. We don't know who did it, but this is a complete misnomer. So somebody called it this protein, so the name kind of got stuck in the literature. We can't do anything about it. And this gene sort of led to a small friendly competition between the two stars of neuroscience back then, Tom Sudoff and Morgan Schenk, each having completely different idea about this gene and what it could do. So till now, there's no disease. Everything is actual science on the gene. And what Tom always thought that it is a synaptic protein. So all his papers are with synaptic, here again, synaptic impairment. I also kind of contributed as a neurexin kinase and so on and so forth. Morgan did not think so. Morgan always felt that this molecule translocates to nucleus and is a transcription activator. So this is one paper and this is another paper by this group. And essentially, these are the two competing theories about what Cas gene could do back then. And the science community was missing something extraordinarily happening from the other end of the spectrum, from clinical sciences. And that is that soon after Tom found Cas in mouse, there was this paper in 1998, relatively small paper, where Cas gene was identified as a locus for X-linked atrophy, optic atrophy. And in 2007, nothing happened from then. In 2007, when this paper came out, where they were looking at copy number variant in X-linked intellectual disability, and Cas kept on showing. So this paper was very important in my mind, but just a year later, this Nature Genetics paper came out, and they kind of showed with multiple cases that mutations in Cas could actually associated with microcephaly and pontocerebellar hypoplasia. And they completely sided with Morgan's idea that Cas must go to nucleus and help in development. So Cas started as a gene in development and then got embedded as a probable gene in a developmental disorder. And Rett syndrome started as a degenerative, sort of early degenerative condition and then converted back into neurodevelopmental disorder. So these are the two different stories of very similar conditions. And this is one of our work. There are other people who did similar things. I'll show you. You can see how hypoplastic cerebellum and the brainstem is there. The kid is also microcephaly. What we did was carefully took clinical histories and looked at the occipitofrontal circumference from childhood onwards. And it was clear that, at birth it is more or less within the, what would you expected for age. But at a later age, by 30 months, they are clearly falling below 3 standard deviations to standard deviation. So it looks like progressive microcephaly. I'm not a clinician, so I did not have any guts. So I was scared of calling it that. But someone did. In 2012, I saw this paper came out from this group, Diana Rodriguez, Berlin, and they call it progressive microcephaly. So with CASC also, you have the progressive microcephaly, which you see with Rett syndrome. And now we know the Rett syndrome can happen because of mutation in multiple different genes, FOXG1, CDKL5, and so on and so forth. So at that point of time, with Rett or I would have identified Rett syndrome, they would not have known which gene they are working with. They would just have known the syndrome. So we have multiple such conditions where we call postnatal neurodevelopmental disorder, including MIC-PCH, where based on different approaches in laboratory, we have come to the conclusion that most likely they are neurodevelopmental disorder. And for CASC, it was expected because of Morgan's work that without CASC, neurons won't go to the right place during development, and because of that, you will have the condition. And that is what was published in this Nature Genetics paper. So we pursued our work also in the mice, right? Try to figure out what we were really looking at, but with an open mind that this is mice, and we have to rely on it only as much we can. So we collected a lot of case histories. We had at least 50, 60 case histories by the time we started working on the mice, and we are trying to see what happens. We got lucky. We got lucky because unlike MECP2, CASC is very important in mice. So without CASC in boys, most likely you will see pronounced epileptic encephalopathy and death, right? So in mouse, when you knock out CASC, the mouse cannot live. Within 2 hours of birth, the mice will die. So that was a good sign. And now if you look at the female, which is heterozygous, we cannot have a heterozygous male because it's X-linked, but female, which is heterozygous, when they are born, their brain size is normal. But this is postnatal day 75. Few days later, what you will see, the brain size diminishes. So it's clearly not something, a developmental condition that happened in utero, and it is basically becoming small only ex vitro during early childhood of the mouse, if you will. And if you see, the cerebellum is also very small. So it looked like our mouse model was phenocopying the C1 case history that we got very well. And the cerebellum was disproportionately affected. So if you look at the hippocampus, this is the heterozygous mice, wild type, it's almost similar size. But the cerebellum is really, really small. And if you see what you will closely, you will see this is the Purkinje cell layer, this is the internal granule layer, and this is the molecular layer. You see the granule cells are all very depleted, very few granule cells. So because of reduced granule cells, this cerebellum is very hypoplastic. We did not see any evidence of mislocalization of neurons or any migration defect that one could find using this technique. However, we went ahead and tested whether there was some defect that we are missing. So granule cells, in internal granular cells actually are formed outside, which is called the external granular layer, in early infancy. And then they migrate inward. So what we decided to do is get mice which are younger and look at the external granular layer to see if the granule cells are well formed, and then we can migrate better. So this is the cerebellum of a post-natal day 5 mice, which is very young, and this is the external granular layer, this is the Purkinje cell, this is the internal granular layer. What you can see that in the heterozygous mice, which will eventually have a hypoplastic cerebellum, the formation of granule cells are not affected. And since eventually all these granule cells are found inside, even the migration is not affected. So we already begin to see here that most likely what we are looking at is not a migration defect or developmental defect. And in our mind, it's not also synaptic because the number of cells are reduced. It's not that synapses were reduced or there was a synaptic defect. The numbers of cells were actually reduced in granule cells. So we wanted to first understand what is going on, what is the molecular basis for such a disease. That's something that a question that everybody asks. And I kept asking myself, and whenever I talk to anybody, my last psychiatry grand round I gave, somebody told me you should look at the RNA-seq or data to see if RNA changes. I personally do not believe RNA-seq tells you anything because it's an intermediate between proteins and their expression change does not tell you that the protein is altered or something like that. It's a change that is important, but I don't know how to interpret it. However, I went ahead and did the experiment. Surprisingly, when you do the RNA-seq data, there is not many changes in RNA. This is all. That's all number of RNA that got changed. So clearly, Cas is not involved in transcription activation, as one would have expected. If it were, you will see large number of changes. The biggest change we saw was actually in extracellular proteins, and all of them were increased. And that's not a surprise given you have hypoplasia and microcephaly. When you have conditions like this, the extracellular matrix is remodeling itself continuously, and that is what you will expect. There are some nuclear changes. Most of them are not transcription factor. We think that might be compensatory. There are a lot of changes in solute carrier and ion channels. Here, that shown here. We found some very surprising change in genes that are not even nuclear. These genes are actually coded by a genome that is present inside mitochondria. And that is somewhat surprising to us. So why is the mitochondrial genome getting altered in its expression? So what we did, because as I said, I personally think we should look at the protein as a molecular change. So we took similar brain and did whole brain proteomics to see which protein changes. That might give us some indication of what is going on. When we do that, indeed we find 500 different proteins that are altered, that are either increased or decreased. And if you classify them, there are 3 main classes. One is protein biosynthesis, the other is synaptic, but the largest group by and far is mitochondria. So we think is that what happens with loss of Cas, the mitochondrial function is altered, and the increase in the mitochondrial genes that are expressed from mitochondria is some sort of compensation maybe that the cells are having. So why does this thing changes? Clearly there's no change in mRNA, so why does the protein change? So to answer this question, what we did an immunoprecipitation experiment with Cas, and we did not cherry pick. We just laid it out using string diagram to see which groups of protein does Cas complex with. And what we found is that indeed it complexes with proteins present in synapse, as we knew, with proteins involved in protein biosynthesis and as you can see here, and mitochondria. So it's the same complexes, in many cases the same protein that goes up or down, Cas sort of interacts directly with those complexes. So we feel that Cas can interact as a scaffolding molecule with large number of different complexes in neurons, and when you take away the scaffold, these complexes get unstable and changes the function within those complexes, giving rise to this condition. And does it have a functional implication? So the answer is yes. If you knock down Cas from cells, here we are showing knockdown, and here you can see the quantification of the knockdown. What you see if you knock down Cas from cells, the mitochondrial expression goes down. you put back cask into the same cell, and the mitochondrial respiration is restored. So it seems that if you take out cask, indeed the mitochondrial function is affected. Now this is from the brain of the mouse, which doesn't have, which is heterozygous for cask loss. Again, you take away cask, and what you see that oxygen consumption goes down, the respiration goes down. Along with that, glucose oxidation goes down. So bottom line from till here, what we think is that cask is a scaffolding molecule. It might be involved in synaptic functions, clearly involved in mitochondrial functions and protein biosynthesis. But the crazy part here is if it were involved in protein biosynthesis, indirectly it might also affect synapse and mitochondria. So we don't know clearly exactly how it functions. All I can tell you, which proteins it complexes with, and the fact that the mitochondria is actually affected. So if mitochondria is affected, that gives us a clue as to what is going on. Because loss of mitochondrial function is often associated with degenerative disorder and not developmental disorder. So that might explain why we keep seeing that the neurons are born normally and grow fine within utero. You have to understand within the in utero conditions, the mother is providing everything in the placenta, via placenta. Plus, some parts of the blood barrier, brain barrier is still not complete, so the brain gets enough nutrition. And ex utero, the baby has to fend for itself. It has to suckle, it has to do everything. The blood-brain barrier is complete. So at this time, the nutrition that goes to the brain also reduces, plus the demand increases. So this is the time then, if there were a mitochondrial defect, that you will start seeing cells loss. So I just told you that we could not study the knockout mice further, the complete knockout mice further, because the mice died, just like you would expect for a human. So what we did of the cases that we have collected, we started focusing on boys who can be kept alive using medical intervention for at least a period, who have no cask expression. So cask null mutation in boys. Now, this is a mutation which is very early on in the protein. This is a protein, this is a gene in the second exon. So very early on protein, the 27th arginine is converted into a stop codon. So essentially this boy doesn't have any expression of cask. Now this boy would die within next 66 days. This is very early on. So by the day 66, the boy dies. Now if you look at his cerebellum, it's really a thin wafer. It's really small. Look at the brainstem. The brainstem is also very thinned out. Now these boys actually died because of central neurogenic respiratory failure. And how do we know that is central neurogenic respiratory failure? We found another boy with the same mutation, in which in the intensive care unit during his, just after his birth, he got a tracheostomy done and was on assisted ventilation, and that boy lives till date. He's 18-year-old. He's neurodevastated, has no brain functioning in whatsoever, but he did not die. So the death itself is most likely because of neurogenic respiratory failure. And we went ahead and checked the knockout mouse just before it died, and we found its ventilation is very, very reduced. So that most likely is the cause of death. So since we got some cases like this, we looked at their brain activity, how their EEG pattern looks like, and it starts very, very disorganized in the beginning. Type of EEG that could be called either hips arrhythmia or atypical hips arrhythmia. But later on, it takes up this typical configuration where you have this burst of activity followed by near isoelectric lines and then burst of activity again. This pattern of EEG is known as burst suppression and is typically associated with an epileptic encephalopathy condition called Otara Syndrome. Now, these kids would typically get it, and they have multiple seizures a day. They have 10, 15 seizures a day. So you can give like people, some of this, one kid takes, I think, 7, 8 GABAergic drug, but does not help, completely refractory to drug. So they typically get a diagnosis of Otara syndrome, but basically this is happening because of lack of cask. So this kid, as I said, died at day 66, 2 months after his birth. So the parents kind of donated the body, so we were able to, for the first time, look at the autopsy of a child without caste and ask ourselves that we think that it is degenerating. Everybody is telling us it's developmental. What is it really? So yeah, indeed, the brain is small. It's microcephaly. So you see, this is expected for that age, and this is what we found. So indeed, the brain is small. But look at other organs: liver, pancreas, thymus, kidney, adrenal. Everything is small. The entire baby is small. The baby has a somatic growth retardation because of loss of caste. So although you expect the brain to have, you have for brain there is certain degree of freedom in growth retardation. You don't see brain getting small because brain is spared. There is brain sparing effect. But part of it could be explained for by the just smallness of the baby itself. But if you look at closely, you can see tertiary gyri. A tertiary gyri develops during the final weeks of just before birth. So it's not that developmental was wrong. The tertiary gyri developed. There is no problem in the configuration of the brain. Of course, if you look at the cerebellar hemispheres, they are very, very small, very tiny. And the brainstem is, of course, very tiny also. So, a general impression that we got was that it's a small brain. The whole body is small. And the brain, if I just take a look, forget it's small. The only thing I could tell you from here is that the cerebellum and the brainstem is very hypoplastic. But it did not look as a problem of a development at back then. So we looked at the histopathology of the cerebellum. We got another child of similar, we could not get exactly 2 months. So this child is around 45 days old. She's a girl, so it's not also the same sex, but died of unrelated cause. She died of pneumonia. So if you now compare the two cerebellum, what you see, of course, you have the external granule layer well defined in the girl because she's younger. The migration is not as much here. But here you can see the migration from external granule layer to the internal granule layer in this mutant boy, this boy with the mutation happening. The Purkinje cell layer is well lined up. So you can see the Purkinje cell. So we could not see any problem with the arrangement of the cerebellum also. It's just small. And if you look close by, if you look at any area and you compare number of cells in the external granular layer, internal granular layer, or Purkinje layer, or thickness of the molecular layer, the configuration was not wrong. It's just that it's very, very small. So one way of thinking for us back then, what we started hypothesizing is that, yeah, it may be a degenerative condition, but the degeneration happens even as the neurons are migrating and developing. So if it's very early on, how do we separate? We can't separate. And the problem here was looking for neurodegeneration is that these brains were obtained by standard post-mortem. So by the time we get the brain, it was not, the cells were of course always dying in both the conditions. So we could not really look for neuronal death markers with molecules for neuronal death. So what we know that if you have neurodegeneration and neuronal loss, then astrocytes tends to react with astrogliosis. So astrogliosis and gliosis is a marker for neuronal damage and neuronal loss. So we said, let's look if there is gliosis going on. Remember, in Rett syndrome, they found some gliosis. So we asked ourselves whether in this condition also we see gliosis. So we went ahead and immunostained the brain with a marker of gliosis called glial fibrillary acidic protein, GFAP, and asked ourselves if there was reactive cells. So this is the girl who died of unrelated condition, cerebellum. This is the boy who died of loss of caste. And look at the granule layer. The yellow is the GFAP stringent. The gliosis amount is 5-fold that of the other condition. So it's huge amount of reactive astrogliosis. In some sections, if you look close by, you can see this bunch of astrocytes sitting in the granule layer, which are all activated and expressing huge amount of GFAP. So this gave us a strong clue for the first time in human, coming to a complete circle, which RETT did in 1966 for Rett syndrome. We ended up doing for CAST now. And yeah, here also we see gliosis, which is a marker for neuronal loss rather than a neurodevelopmental disorder. So the next set of experiment for us had to be conclusive because we wanted to make sure, we don't want to make a mistake here. Can we separate then the neurodegeneration from developmental stage? We can't do this in human who have the mutation from embryonic stage, but we can now try and design a mouse where cask is lost, but not lost in the embryonic stage. But cask is lost in set of neurons after the neurons have developed and have migrated inwards, right? To do so, we tested a lot of different ways of doing, and this is using an experimental setup called Pre-LoxP system, where we have this cask gene. This is the exon 1 of cask gene, which is FLOX, that is, have LoxP sequences on both sides. Now, this LoxP sequences in presence of free recombinase will recombine, and that will lead to excision of the gene. So if we make sure in which cells the free recombinase is expressed in a mouse where Cas gene is FLOX, we can delete Cas in a temporal and a spatial specific manner. So it will be spatially and temporally controlled. And we first did this because we don't have very good antibody for Cas. We first tried to do this experiment with the indicator mice, where depending on where the free recombinase is expressed, those cells will turn fluorescent. So we went through a variety of mouse lines, and we homed into a molecule called calretinin, and free recombinase is expressed under the promoter, which is driven by calretinin promoter, and looked at its expression in cerebellum. And this is not red, this is yellow, but this is the fluorescence essentially. On postnatal day 8, you hardly find much expression in cerebellum. Most of these cells are Purkinje cells, which are already there, and they are expressing. So a lot of Purkinje cells are already expressing this Cre recombinase. So by day 8, Cas will be excised in those cells. But note here, there is this is a nucleus, saying there is outer layer of blue, which is the external granule layer, where the granule cells are forming. Look at that. That has no expression of yellow. So that means when the granule layers are birthing and they are migrating, it doesn't express Cre recombinase. Okay, they will still have Cas at that stage. Cas won't get deleted. Now, if you wait a few days, this is postnatal day 15, a week later, you see the Cre recombinase is expressed in olfactory bulbs, striatum, hippocampus, but of course in cerebellum, which we are really interested in. Now look again here very carefully. Again, this is the external granule layer. This is the internal granule layer. In the external granule layer, there is no expression of red. But the internal granule layer, you start seeing the red. So what is happening here when the cells, either be the Purkinje cells or the granule cells, when they develop and now they got to the right place, they lose Cas. So now with this experiment, we have separate loss of Cas from the developmental stages of neuron, and ask ourselves what will happen to a mouse once we separate it out. Can we now see the degeneration? If now we can see degeneration, we can conclusively say this is a degenerative gene and not a developmental gene. So having this tool at hand, we went ahead and tried to figure out. First, we were very disappointed. We did not think we have gotten anything. The mouse looked absolutely fine, even though Cas was deleted from cerebellum by day 15. It kept on developing in right trajectory. It looked healthy, sleek coat, adequate nutrition. But we waited. We were patient, and we were rewarded. This happened on the 100th day of the mouse. Now you will see this mice is able to take a single step without problem. Not a single. He has absolutely no balance, no motor coordination. So what we did, can I remove this box somehow? Okay, I'll start it again. So what happens is that we take out the brain and ask ourselves what is wrong with this brain. And what we found is that this is the normal mice on day 100. This is how the cerebellum should look like. Cerebellum is very, very small. But the question comes whether the cerebellum was always small and the mouse still learned to somehow live with the small cerebellum and then lost its function, or is, can we actually show that this is a degenerative condition? And we managed to get a conclusive evidence for the first time. What we are looking at are degenerative conditions. So now we are looking at the mouse, which will eventually have ataxia and incoordination. But we are looking at an earlier time, that is post-natal 13, that is 15 days after Cas was deleted, and ask ourselves what happens. And you see the cerebellum was completely well formed. The same mice, not same mice, but same genotype mice, in another 70 days, you see how small the cerebellum is. The cerebellum without Cas develops now properly. The functional is there, and then only after the development will start degenerating, and the functional loss that we see in Cas is only dependent on the degeneration. Without a degeneration, you won't see a problem. So it's not synaptic, it's a disease of degeneration. So you can see how small it gets, and this part area, which is supposed to be the white matter, seems to have been expanded, and the cells you see, the granule cells are almost totally gone, thin layer remains. And now if you look at the cerebellum, the cross sections are very small, molecular layer is thinned out, granular layer is depleted, and this white matter area seems to be somewhat increased. So here we have an evidence now for X-linked gene, which is considered to be neurodevelopmental, showing that it's actually degenerative. Now, as I showed, that mouse has a lot of locomotor phenotypes. So it's almost, because they can't walk, it's almost painful to do any kind of locomotor test. You all don't even want to, keep them alive for very long. So it's just not humane. So how do we show that the locomotor loss that we see is not coming from some synaptic defect, but is actually a function of the atrophy itself? So here we have done an experiment where we put the mouse on a rotor rod, ask them to balance. This is on day 48. Now this is reduced from the control, but still it's not significant. Look at what happens from day 48 to day 70. In 22 days, the mouse completely loses its ability to balance. So the functional loss that we are seeing is the loss in the physiology we are seeing is a function of the atrophy, is function of the neuronal loss, and not synaptic function or some molecular function or some kinase phosphorylating something. Those things are important, but what we are really saying is neuronal loss. So just to make sure that it's not synaptic, because I come from Tom's lab and I've been studying it as a synaptic protein for 10 years before this, or 12 years. So I asked myself whether we see loss of synapses. So we stained the cerebellum with a molecule called bazoon. And we don't see a change in number of puncta which are synaptic, which is somewhat confusing to us. If there were cellular loss, wouldn't you also see a loss in synapses? So why don't we see loss of synapses? And one of our ideas is that neurons outside of the cerebellum, which form mossy fiber connection with the granule cells, are now forming it with the Purkinje cells which have not died. So synapses are still there, but the cells are lost. But the synapses are wrong. They are making a wrong connection because the cells that they typically make connection to is lost. So we don't think there's a problem with formation of synapse or anything like that. It's clearly what we see is a degenerative condition. Now remember, I said that the white matter area is enlarged. So this is another example of another pair. You see how big the mouse cerebellum is on day 30 and how small it becomes by day 100. It's very, very small. And this is the white matter layer. Now we have stained this with fluoromyelin, which stains myelin. But we don't see much white matter there. So white matter is also actually reduced. So only the space has expanded, but it's not that it's full of axons which are myelinated or something. So although we call it white matter layer, there's nothing white remaining there. All that is there is the extracellular matrix. And that explains why we saw an increase in mRNA with extracellular proteins in our RNA-seq. Because number of cells which makes the extracellular matrix have decreased. And it has to maintain the extracellular matrix, so it makes more and more. That's all we are looking at. It's just an effect. And finally, as I told you, that astrogliosis is one of the prominent markers for neuronal loss or cell death and neurodegeneration. We saw that in humans. So we asked ourselves, do we see that in mouse too? This is a mutant mouse, the cerebellum is degenerated, and this is the normal control mouse, and you can see that astrogliosis is very increased. So with this, what we have now, I think this paper is now submitted. We are waiting. It's been a very hard thing to publish. The hard thing to publish because we ourselves kept calling it neurodevelopmental like everybody till 2 years back, and now we are suddenly trying to say no, it's neurodegenerative. Now we have evidence. We are showing that till 100 days is fine. But like anything else, science is also somewhat dogmatic. You have big leaders who think that this is how it has to be, and the people are surprised when we ourselves are surprised. Because we let the mouse go, and our first, if you see one of our talk I gave 2 years back, I said CAP is not important after development. Mice is fine. Only once the mouse started falling and gave us a surprise did we come to know that we are looking at degeneration. Now this degeneration takes a lot longer time. Remember, than the cerebellar hypoplasia, which is very quick. And we think that it takes longer time because cerebellum is more mature, there is more glial cells to support the neurons, so it's just more steadier. So it takes longer. So how does this X-linked disorder then masquerade as a neurodevelopmental non-progressive disorder? Why is it? So the idea of neurodevelopmental is that's always considered to be non-progressive. Here is the reasoning for that. Now, X is basically 2 doses in females and a single dose in males, right? Now, in typical condition, there are 2 X genes on 2 different chromosomes, and in any given neuron, one of these genes will be randomly inactivated. But since both these chromosomes have normal Cas genes, all neurons will have Cas, and the cerebellum and everything will be fine. There's no problem. Now consider this, that all these girls who have this developmental disorder are heterozygous. In a typical heterozygous condition, this red, consider this red X chromosome, which has a mutant Cas gene, right? And then you have this gray one, which has a wild-type Cas gene. So this will go random inactivation, creating a mosaic scenario in a brain of the girl. You will have 50% neurons which have Cas, and then 50% neurons which do not have Cas. So even if the degeneration is going on, it will plateau at certain stage because 50% of neurons still have Cas. And that causes the plateauing and non-progressive nature of this disease. And then the males, where it's lethality, where you have this pronounced, profound neurodevastation and encephalopathy. You have mutation in a single Cas gene. You can mimic that in mice by creating a homozygous condition in lab animals. And in such condition, there won't be any Cas in any of the neurons, and that will lead to rapid degeneration and death. So what is the bottom line? The bottom line is, based on our studies, we suspect that there are multiple different genetic conditions like Cas mutation and Rett syndrome. These are pathologically neurodegenerative. Mind you, they will still fall under the policy definition of neurodevelopmental gene because it's still happening during developmental phase of the child. However, due to random X chromosome inactivation in the heterozygous mutant girls, 50% of cells still express the functional gene resulting in a non-progressive pathology, whereas complete loss of the only allele in boys lead to unconstrained degeneration and encephalopathy. This is what we really are observing in this condition. What we are saying is that essentially we are agreeing more with early clinical and pathological impressions of Dr. RETT, and I think our work on mouse sort of misled us and drew into a wrong area. Now, is this a bad news for people whose children have this condition? I don't think so. Of course, right now, yes, but I don't really think so. The reason I think is somewhat positive, or I'm optimistic about it. If these are postnatal conditions where the degeneration is happening after birth, we have a window period for intervention. Then all that we need to do is come out with a way where we can actually stop the death of the neurons after the child is born, and we can halt the disorder even though we can't reverse it. We can at least halt the progress whenever we find it. So I'm sort of optimistic about our findings, and I think this will lead us to good things. And we are starting a study. We have some ideas of what to do to prevent neuronal death. And we are starting some of the studies, and we think if we could do that, it would be great. In some cases, we do hear case histories where the girl is otherwise fine till 6 months with Cas mutation, and then starts to sit even starts to stand, and then they develop epileptic spasms, and they show signs of neuroregression, and they lost all acquired skills, just like Rett syndrome. Recently, I heard something like that from a girl who is now 9-year-old. At 2 years of age, she was standing, and now she cannot do anything because of these epileptic spasms. So what I believe in such cases are happening is that there is a unfortunate or unfavorable skewing of X chromosome towards the mutant gene. So in those cases, if you have, say, 30-70 or 80-20, so 80% of neurons might not be expressing the normal Cas gene in girls, and that is leading to degenerative condition, which looks like a neuroregression. This is where we are at now. And that brings me to acknowledgement. These are lab members who have contributed greatly to our study. Brushali, she is now gone to New York. Jeffrey has become a musician. Chen is now doing his postdoctoral fellowship at Baylor. Leslie, you might know. Paras is a grad student right now in the lab. I have collaborations with Dr. Michael Fox and Dr. Sarika Srivastava at FBRI. And the funding source has been National Eye Institute, Virginia Tech, and National Institute of Health. With that, I can take any questions. Yeah.
For trying to intervene and stop the neuronal death for those conditions, I would imagine that, you know, these kids develop normally early on. I would imagine that you'd really want to intervene much earlier, so like before you start to see the symptoms, but you're not going to make the diagnosis until you start to see the symptoms, at which point do we know how much of that degeneration has occurred? Is it 50%, 90% by that point?
Yeah, so that's why I say that you basically can do only half, and really can't reverse. Because by their very nature, these are genetic disorder but not inherited disorder. Typically, most of these cases are de novo conditions. Because this happens in girls, and boys would be dead, it's impossible to have them as inherited conditions. So, yeah, so diagnosis is made only after the symptoms have arrived, essentially, and initially even pediatricians miss it till they find that they are having progressive microcephaly by recurrent measurements. You are absolutely right. We won't get to stop the degeneration before it starts, but it takes a time. There's still several months to years where we can stop it. So our whole idea right now, how to go after such condition, is two-pronged. So I have one collaboration here with Stephanie DeLuca's Neuromotor Clinic, where we are testing if intensive therapy in early childhood, just after you find, would reverse some of it functionally, if not structurally. So if your cerebellum is defective, you have no central axial tone. So if you could actually give back the tone to some extent or something using rehabilitative therapy, together with a drug that will stop further progression, it will improve the quality of life tremendously. These are clearly not cured, but improve the quality of life tremendously. The second thing I feel is that these girls lack the essential homeostatic mechanism that all of us kind of take for granted. When we go around, we are always exposed to neurotoxins, infections, stress outside, but because of homeostasis, our brain is not giving up. But many of these girls don't have that luxury, and simple things can give rise to neurodegeneration rapidly later in life, even in adolescence. So if you have a drug that could stop the neurodegeneration, we could at least intervene even those points where there's some kind of stress which is causing neurodegeneration. So yeah, I'm not saying that we can reverse that. And since again the mutation will be detected only after symptomatology, and they are all de novo, they are also not some real good candidate for any gene therapy or something.
So we talked about this being de novo, and this is X-linked. And in your models are mostly mice. Translating that into humans, you mentioned 2 patients who were males, who one was trached and invented, and 18 years later, I think you said it was still living.
Yeah.
The one boy died. So is that the typical course, that they die as in the neonatal period, or is this an in utero death and considered a miscarriage, or because they wouldn't be that, because you said in the utero they develop okay.
Yeah, but I am sure there are a lot of miscarriages also, because counting cells in brain is like counting stars. To try is difficult. So we have taken a proxy of the central nervous system in retina, because there you can count number of retinal ganglion cells in mice. So what we find that this is a disorder. So it's weird. So in mouse, the development is different than human, as I said. So mouse, when it's birth, basically when it's born, till day 10 of after birth, it is similar to the third trimester in human in terms of development. Only after day 10 can you consider in terms of equivalence of our Carnegie staging that it is ex utero stage, after birthing stage. So when we actually look at when the cellular loss is in mouse and equate it or extrapolate it to human, we find the cellular loss starts in the third trimester. So yeah, it could be early on. It depends on which countries you see. In most countries still don't have whole exome sequencing. We don't know how many cases are there. A paper just came out yesterday out of Italy. They started exome sequencing on pontocerebellar hypoplasia, and they found that CAS in Italy at least is the commonest cause of pontocerebellar hypoplasia. So I am sure it happens in other countries also. In countries like India, where nutrition in periphery is already poor, I believe we'll have lots and lots of low birth weight and immediate death that we just don't even count. And that might be true for MECP2 CAS. So it's a problem of diagnosing and also keeping track for boys. I think that's why initially what we thought is that most of the cases that we see are girls. So in CAS mutation, almost all. Most typical cases are girls. Now, sometimes it's a boy. So if it's a boy, typically you see a missense mutation because that's a hypomorphism, not complete loss of function. And in some cases, it's mosaicism because it's a de novo mutation. It can happen, somatic mutation during embryogenesis. So suppose it happened at 2-stage or 3-stage cell stage. Every third cell is CAS or second cell is CAS knockout in the boy, and it mimics like the girl in that mosaic condition. So such cases are also there. And we now have found a truncation mutation in boys who are 8 and 11 years. We are writing a paper on it. And this is the most fascinating case I've seen till now, and we have got an explanation for that. So these boys have a truncation mutation which makes the protein dysfunctional. But the boys didn't die, and their cerebellum is completely normal. And we were like going crazy. How is this happening? Till we figured out that this point mutation is happening in a evolutionarily novel exon, which is not expressed in the hindbrain. So that exon is spliced out in hindbrain. So even if there was a missense mutation in that exon, the hindbrain never sees that mutation. So for it, the entire protein is formed. So hindbrain is fine, but the forebrain makes this exon, and the forebrain doesn't see CAS. So the boys have microcephaly and intellectual disability, but no cerebellar hypoplasia, and it's really, so how we were wondering what happened and what stage, and what we find that the exon is not even expressed in hindbrain of mouse. So it must invertebrate very early on. That exon must have taken on some function that we don't know yet, that is very specific for COVID. So it's really cool, and that's why I think it's very important for us to collect these case histories and then match with mouse to make sure that by Rett syndrome we don't go on a wrong track.
One last question. So we're clinicians. You're obviously a basic scientist. So how, in our partnership with Fralin Biomedical Institute, how can we work together to help to support your research with the patients we see?
Okay. So we have a very good example going on right now. Actually, I'm part of it, and I'm not a PI on it. But it's very interesting. So this started with Dr. Otara in neurology when he was here. So what he found was this particular case that came in, and he had It's not flapping, it's not asterixis, it's not a flapping tremor. It's some sort of tremor. I can't describe it. It's a very coarse, low-frequency tremor of hand and some gait issues. So he got an exome sequence from that person, and it turns out that the person has a mutation in DRP1, which is an important protein involved in mitochondrial fusion. So he contacted Dr. Srivastava here, and what she did was ask them to do a skin biopsy on the person and another one, and she established 2 different cell lines, and she has some fantastic results now showing what happens with that mutation. It's exactly reverse of what would you have expected, and it's kind of fun to see what's going on there. I am just right now working in periphery, and what we are doing now is taking those fibroblast cell lines and we are converting them into neurons using induced neuron technique, and what we'll see how those defects that we see in normal mitochondria translate if it was neuron. In neuron, the dynamics of mitochondria is even more funnier because it has these long stretches of axons and dendrites, and the mitochondria has to do its own trick to go to right places in the right way. So we have not gone there yet, but we have started making neurons out of this fibroblast, and we hope to see some. So does that answer your question?
It does, thank you.
And sometimes even if we, in many cases, in this kind of cases, even if we get just a mutation, we don't even need skin samples. We may mutate the gene in lab and ask ourselves what goes wrong. And in this particular case, I was just telling you in the end, we have no other information, just information from this case. We have no biological material. But since we have the mouse, we could ask ourselves whether the exon expression were different in hindbrain or in forebrain, which we can never do from humans.